Hi, my name is Jonathan Rose, and I'm going to talk about my favorite S&P thing, which is phenomena associated with center surround receptive fields, specifically visual receptive fields. And to understand this concept, we're going to need to look at kind of the basics of the visual cascade before diving in, which consist of photoreceptors, bipolar cells, and ganglion. It is important to note, though, uh, that many bipolar cells can innervate one ganglion cell, and certainly more than 3 photoreceptor cells uh, can connect to one bipolar cell. The cascade starts when stimulus hits the photoreceptor cells, leading to hyperpolarization and a sequential cascade from the photoreceptor cells to the bipolar cells and sequentially down to the ganglion. And this is where the receptive fields kind of come into play. Anything connected to one ganglion is considered one receptive field. And within that receptive field is broken into 2 subgroups. So one in which there's a center and those that are surround. And these refer to certain photoreceptors peripherally to the ganglion. Now there's 2 types of receptive fields. One called an on-center off-surround and another one called an off-center on-surround. So an on-center off-surround refers to anything that stimulates this center will cause an excitatory response. However, any stimulus out here will cause an inhibitory response. So if light only hits the center, you're only going to see excitation. If light only hits the surround, you're going to see inhibition. And it's vice versa for the receptive field over here. And this can lead to some really interesting phenomenon. One includes the Hermann grid illusion. So if you look at the intersection of these white grid lines, you'll notice that they seem quite a bit darker than the grids themselves or outside the grid. And in fact, if you sample this spot right here and sample this spot right here, they are in fact the same color. They're both white. So why does this area seem darker than this area? And this is where these center surround receptive fields come into play. So this is an on-center, off-surround receptive field. And kind of review again. The on-center means any light that hits this area is going to cause excitation. Any light that hits this area is going to cause inhibition. So the off-surround that's in this black area isn't going to cause any inhibition. It's null. But the light that hits the center here is going to cause excitation. So you're going to see complete excitation and a little bit of inhibition, which is noted by these grid lines here. This is for excitation, and this is for inhibition, and your overall excitation in gray. However, what's slightly different is if you put this on-center off-surround receptive field the center of these grid lines, you'll see that there's a lot more inhibition than there is excitation. Same amount of excitation, but more inhibition, making this area seem slightly darker than this area. And this also can be seen in another illusion called the scintillating grid. So if you focus on this area right here, you'll see this appears to be a white spot. If you kind of look in your peripherally and look around, you're going to notice that black dots disappear and appear in these white circles. But anytime you try to focus on it, they will disappear. These spots are in fact white. So this is an illusion. So behind this illusion is the center surround ganglions again. There's an on-center, off-surround ganglion receptive field. So you're seeing excitation of the on-center, which is seen here, and you're seeing inhibition right here, but not in these areas. So you're seeing an overall gray color. And when you look in this area, you're receiving more excitation because this area is much more light stimulus. However, you're also seeing more inhibition. In fact, you're seeing more inhibition to excitation than you saw previously here. As you can see by the gray line, which indicates the amount of excitation. So you're seeing less excitation, making this appear darker. Now this doesn't occur when you are directly looking at it because your fovea has much smaller receptive fields, so there are a lot more smaller fields within these white dots than one large one like this. But in your peripheral vision, you will see black dots appear, and as you look towards them, your fovea, who has a lot smaller receptive fields, will distinguish them as actually white dots. So that's the end of my presentation. I hope that it was informative, and here's the citation to where I got the final grid line from.